Metropolis. The COVID-19 pandemic caused by the SARS-CoV-2 virus continues to evolve globally, with the most recent development being the availability of vaccines. Vaccines help us by protecting against the SARS-CoV-2 virus infection by inducing specific antibodies. We at Metropolis have launched a robust quantitative test for antibodies to the SARS-CoV-2 spike protein named COVIPROTECT. Why should you use COVIPROTECT? COVIPROTECT Test quantitatively measure antibodies if you are exposed to the SARS-CoV-2 virus or are vaccinated. It is unlike the other qualitative assays available in the market. This test targets antibodies directed against the particular region of the viral spike protein responsible for binding to the host cell receptor and entering the host cell. Most current COVID-19 vaccines, either approved or in development, induce antibody response against the viral spike protein. COVIPROTECT test helps to identify your exposure to SARS-CoV-2 virus, measure your antibody response to vaccination indicative of effectiveness of a vaccine. Timing of the COVIPROTECT test. There is a significant inter-individual difference in the levels and chronological appearances of antibodies in COVID-19 patients, but median seroconversion has been observed at approximately 2 weeks post-exposure. It is advisable to test for the neutralizing antibodies after 21 to 28 days post-COVID-19 SARS-CoV-2 infection and 15 days post the second dose of the COVID-19 vaccine for detectable levels of neutralizing antibodies. How will you interpret the values of the test? The test is reported in international units, ml. Value reported quantifies the antibodies present in the sample. If value is below the cutoff mentioned in the report, it indicates absence or very low levels of neutralizing antibodies, IgG, to SARS-CoV-2. Values above the cutoff indicate the presence of neutralizing IgG antibodies to SARS-CoV-2. It generally indicates exposure of the subject to SARS-CoV-2 due to infection or vaccination. Values at cutoff may be equivocal and a second sample should be collected, preferably between 2 to 3 weeks after the first test. Unique features of the COVIPROTECT test includes highly sensitive 98.8% and specific 99.5% assay, excellent positive agreement rate to neutralizing antibodies PVNT, which protect against future infection with SARS-CoV-2. Approved by regulatory bodies like CE and US FDA as emergency use authorization. Done at NABL or CAP accredited laboratories of Metropolis following excellent quality standards. Useful for assessing the current vaccination. Faster turnaround time with same-day reporting. Things to remember. Results from antibody testing should not be used as the sole basis for the diagnosis or to exclude SARS-CoV-2 infection or to confirm the infection status. Your doctor will interpret the results in conjunction with other data, example, symptoms, other test results, and clinical impressions. Detection of neutralizing IgG antibodies against SARS-CoV-2 indicates the presence of protective antibodies in vaccinated individuals. The number of antibodies or the duration of antibodies that will protect against infection remains to be determined. Get yourself tested with Metropolis COVIPROTECT to know your exposure to COVID-19 or your antibody response to vaccination.